Welcome to this short video on natural language processing, particularly with a view to protein language models. This is basically a video for beginners on this course, B10 Python for AI. So let's talk about natural language processing, and this is used for a wide variety of tasks, including text cleaning, vectorization of that text, training a text classifier. There's a number of steps associated with it. The first of which is cleaning and tokenization. That's basically removing punctuation, making everything lowercase, splitting the text into words or suburbs. And there's different Python libraries that can use to do that. LLTK and Spacey are just examples of that. We also want to identify stop words and stemming. So we want to use common words like the, is, and that have limited meaning. And stemming also means that we simplify words that go from running, for example, to run. So it's trying to make them less more systematic across the whole text that we're looking at. Then more steps. We have vectorization. That's so one of the key steps is used to use term frequency inverse document frequency, TF-IDF, and Bagger words to convert the text into numbers that a model can understand. So the text is abstracted in that format. This gives a high rate to words that are rare across documents, but frequent in a specific one. These words create embeddings. So the words are represented as dense vector, where the words that are similar sit close together in space. So laptop and computer, puppy and dog, kitten and cat, for example. So let's also talk about transformers. So transformers are key to protein language models. So before transformers, we had recurrent neural networks, and they're difficult to parallelize. So they have to take one word at a time, going from left to right, and therefore long-range dependencies. So for example, if you know the German language, the structure is quite complex where often the verb is at the very end. Or if you've seen certain films where someone like Yoda is speaking, the way in which language is communicated means that you have to keep track of each word as you go through a sentence. And this creates issues that we have no parallelization for these processes. An example of this, if we look at this is text from Ulysses, you can see that the while there's lots of texts that have a particular pattern or a particular way of communicating, when we think about, for example, creative writing, it can be more difficult to think of systematic ways in which to organize that, and certain models may perform poorly with, novel, unexpected ways of expression. So our transformer, in more simple terms, is basically trying to change "Je suis étudiant" to "I am a student." So taking this French text, abstracting it, and therefore what we refer to that as encoding it, and then decoding that abstraction into a different language. So there's a transformation from the left to the right, and that transformation is basically numerical. So it's a numerical operation where we abstract the item on the left into numbers that are changed, and those numbers are decoded then on the right to produce these letters in English. So the input text goes in here to an encoder to abstract it. It goes across to the decoder. It's decoded, and we get our output hit there like that. There can be many layers of encoding depending on the complexity of what we're looking at, and same with the decoders. And all of these are typically structured in the same way. So for an encoder, we might have two layers present. One is the self-citation layer, which basically helps the encoder to look at other words in the input, and that as it encodes a particular words, and then a feed-forward neural network that helps specify the words that are more important in that sentence. For the decoder is three layers, a self-attention one, and a feed-forward neural network, and then the encoder decoder attention allows focus on key tokens in a sequence. So what's key about transformers in terms of protein language models? It simply wouldn't be impossible without them. So if we use the old methods of recurrent neural networks, it would take too long. So transformers can do all the tokens at the same time. So we might be dealing with amino acid sites or text. Um, each token can the context of each token can be considered in the same examination, and the long range context can be retained. This allows parallelization, so the dependency is very much reduced, and this requires a GPU to implement it because it is very, very computing intensive. And this means that transformers can work in large data sets efficiently. One consideration is that we can, a bit like learning a language, you can pre-train on billions of tokens of text and then fine-tune for small task-specific data set. So for example, you might have a generic uh, transformer that has created a model based on a lot of text for writing in French based on English input. And this kind of, I guess it's basically a French, like a dictionary, would therefore could be fine-tuned to work on scientific, English scientific writing to produce scientific writing in French. And obviously how good it is to a French speaker depends on how good the model is. So we want to embed each token into a vector. Each word, or for example, when we're talking about protein language models amino acid, is mapped to a high dimensional vector, which is known as embedding. So these are really what we're analyzing here. We're not looking at the amino acids, we're looking at the embeddings of the amino acids. And these are the positions of these are recorded. But then we keep three key components: the queries, the keys, and the values. So the key queries refers to what am I looking for, the key, what do I offer, and the value, what information do I have. Those are the three key components, and each of these are basically contain numerical data referring to the embeddings above. We then compute the attention scores. So the dot product for each query against all keys is computed, it's scaled, and then if we have a high attention score, that means that that particular token or amino acid is more important. So we think of it like this: we have our input data in X, we have our vector of queries, our vector of keys, WK here, and our vector of values here as well. Or we can obtain the queries, keys, and values by getting the dot product of each. What we then do is scale this. We scale our matrix Q by the transpose of K. We scale that by the vector V to give us our output Z here, and that contains the abstracted versions of the input amino acids. So there's quite a few steps here, and obviously this is quite a high level view of the process. So what we're doing is getting the weighted sum of the values. So each vector value is multiplied by its attention weight, and these values are then summed. And therefore this allows the transformer to identify which tokens are more important in the context of the sequence. We then have this step known as multi-head attention. So these identify different relationship types. For example, that might refer to the syntax, the proximity of different amino acids to each other. And these are projected into the embeddings. The embeddings are transformed that way. Following this, there's a feedback forward network and layer normalization. Each token processes through a small two-layer stage independently, and the residual connections and layer normalizations stabilize these steps. So we can ask ourselves, in that transformer self-attention mechanism, what do the query, key, and value vectors represent conceptually? So do they represent three layers of a standard neural network? Do they represent what a token is looking for, Q, what it offers, K, what information carries, V? Do they refer to quantization, kernel, and variance in terms of signal processing? And do they refer to the input, hidden state, and output of a recurrent neural network cell? Well, it's not number one, because we know the layers of a neural network are typically very, very numerous, and they're not referred to by those terms. We know from signal processing that the two terms used for this are different. and they don't refer to the input, hidden state, and output of the cells. They refer to different features of the token, what the token is looking for, and what it offers to others, and what information it carries. So there are basically three features of each token. So transformer types, we use them a lot, we just don't realize it. So GPT is a generative pre-trained transformer. And if you've used something like Google Translate, therefore you've used these already. So take some sort of input, and this could be, something in French that goes through a transformation and gives us output in English at the end. If we think about protein sequence, we can give it amino acids, it goes to a transformer. There's a prediction of the structure coming out from that, which we can see there. And there's different subtle differences between different models. You've got encoders, decoders, and also encoder-decoders. BERT is an encoder only, very good for classification and understanding. GPT is a decoder only, it's good for text generation, so it's more generative in that sense than BERT. And there's also other options like T5 and BART that are very good for translation and summarization, which I think we use less here for this practical. And all use the same self-attention building block. So this brings us more to protein language models, which are based on transformers and GPT and BERT, and these are the models that have been developed over periods of time. So what we want to do is predict the next amino acids in a sequence based on extensive training data without using multiple sequence alignments where possible, initially without existing structure or chemical info. Of course, that could be added on, but we don't necessarily want to add all that at the start if it's very sparse in the context of our data set. So we're using natural language processing, transformer, and diffusion processes. There's a lot of options there. ESM2 and AlphaFold3 are perhaps the most common ones, but there's quite a lot of ones that are available. And we can go beyond this. We can predict function, protein-protein interactions, post-pathogen interactions. There's many, many things that we predict, not just protein, but also RNA, DNA, etc. Anything that's a biological molecule. So again, just to recap on our protein, our basic protein structures, we have amino acids that make up a protein. They might have asparagine, glycine, phenylalanine, and so on. And that's the sequence of amino acids. But when and these form secondary structures like alpha helicase and beta pleated sheets, and they've also formed for a single protein to fold into a more complex three-dimensional structure, tertiary structure. And these single proteins can form complexes. They might be monomer, dimers, they might be heteromers, they might be more complex structures, and therefore the effectiveness of different models to model these complex interactions is also something that's a continuing area of research. So protein language models, what's our analogy? If we go back to the simple aspects again, that we have these letters that are like amino acids, or letters in our alphabet like amino acids. We have a book, which is like our protein sequence, or pages in the book is our protein sequence. We can read a book to extract a short segment containing secondary structure. We can have sentences within a book that allow us to think about the tertiary structure, and we've paragraphs within books as well. So you think about the different levels as we're building up. So we have our alphabet, words, sentences, paragraphs, grammar, meaning from that. I guess for biology, a lot of it centers around the structure that we get at the end, and that is often key. And sequence data doesn't always give us the full picture of that. One of the key models in this area is ESM2. Evolutionary Scale Model 2 is a transformer which is trained on at least 250 million protein sequences. I think that number is changing a lot over time, so that it's likely that will increase in the future. And what we can do is make predictions about using sequence alignment if initial data is sufficient initial data is available. If that sufficient initial data is not available, then it won't work very well. And what it's thinking about is thinking about the relative grammaticality in terms of the feasibility of actually having a protein like this by comparing the log likelihood of the ratio of the wild type versus the mutant, and then also the semantic meaning score. So the distance between the wild type and the mutant as to whether this actually can it make a structure like this, and secondly, would it have a really real function? So other options in this area is MetaAI's ESM fold, which uses the ESM2 embeddings to predict the protein's 3D coordinates. And it's much faster than AlphaFold 2 at inference. Another one is ProTrans by the Ross Lab. Um, it's a family of protein models, ProTBER, ProT5, and ProtoXLNet, trained on protein sequences. And ProT5 embeddings are powerful for things like specific tasks like subcellular localization. They're protein BERT, and that's a BERT style model based on UniRF 90. It's fast. So, for example, if you want to fine-tune a particular biochemical property without a large GPU usage, this would be a good option. So AlphaFold 2 was created in or published in 2021. It uses transformers and multiple sequence alignment. It uses the multiple sequence alignment of related proteins and the evoformer evolution transformer block to extract co-evolutionary signals from that, predict the 3D coordinators, and create the structure from this. And it was good at single proteins. More recently, we have AlphaFold 3, which uses a diffusion model instead and created the 3D atomic coordinates directly. And this was now expanded not just from single proteins, but to DNA, RNA, and small biological molecules as well. And it's there. So if you want to read more about it, you can just go to the server and see how it works. So the steps in AlphaFold are firstly to take an input encoding and take the input amino acids and the tokenized into a consistent format. We then have the performer, the evolution transformer. This performer block processes the amino acids and also creates pairwise interaction matrices to capture which atoms are close in three-dimensional space. We have a diffusion model. AF3, AlphaFold 3 uses this diffusion model to get the coordinates, starting with random atoms, and then it gradually denoises them based on the transformer's data until a plausible structure is being found. And then from this, we have stochastic sampling. So the diffusion model is generative, it makes predictions, and the variation in these predictions affect the model's uncertainty. So if there's high variation, it means it's a flexible region, whereas if there's low variation, it's probably a well-ordered, rigid, and easy to predict region. And these confidence scores are then referred to as PLDDT and PAE. PLDDT is scaled from 0 to 100, where 0 is low confidence, 0 confidence, and 100 is maximum confidence. And predicted aligned error, PAE, is also a metric that is widely used. So AlphaFold, a question we might ask ourselves then is what key advantages AlphaFold 3's diffusion model have over AlphaFold's 2 direct coordinate prediction? So is it because it runs faster because it skips the transformer layers entirely? Is it because it only works on proteins and ignores ligands? Is it generative? Modeling a distribution of plausible structures around the single point estimate, and thus allowing it to naturally handle flexible regions, or does it use convolutional layers instead of attention? So what I guess the key thing here is that it's generative. It uses the diffusion module to be generative and therefore to test different structures to see how much variation there is in the possible structures that are relevant for the protein. So natural language proteins predict the next letter in the sequence. And what we're really getting from things like ESM2 or other equivalent tools is the probability that they're true based on the letters and the meaning to give us the net effect. Often these are based on UniRF, and what's bear in mind is the UniRF datasets and database information vary a lot. So UniRF refers to clusters created using mmseq2, where each is represented by a sequence that has at least X identity to all the other sequences in the cluster. So for example, if we have UniRF 90, it has to that sequence representative has to have at least 90% identity to all the other sequences in the cluster. If we have UniRF 50, has to have at least 50% identity. And therefore UniRF 90 will be harder to train because it has more data, but it might be more specific. whereas Uni of 50 will be easier to train because it has, well, easiest to use because it is a smaller data set. It is fewer clustered, but may not give us the granularity that we want. So classically, protein language models, we'd have considered other ways in which we can consider, for example, amino acid changes, nonsense changes. These are often called variant effect predictors, and things like polyphen and SNP would be examples of these. Or we use evolution models to calculate the relative rate of ancestral non-synonymous site change versus synonymous site change, the NDS. Um, and in some ways, PLMs are trying to do the same thing, just from a slightly different approach. They're using a pre-trained model from ESM2, and so often they're applying approach like deep mutational scanning. So trying to change each amino acid site as you go through a protein to infer ones that are more feasible versus those that are not. And these predictions can tell us more about the evolution of this protein. So one of the considerations that they can use MSA data to create hybrid models that are more effective. The consideration for that is that a lot of the availability of experimental data is quite limited, and objective accuracy is a tricky one to objectively obtain here, because doing experimental structural biology without using computers is a very, very labor-intensive, challenging task to do. So this is just an illustration of the correlation between the human deep mutational scanning and the prediction accuracy for different models. What we can see, VESM is generally doing better, and ESM3 struct less well. What you can see as well is that there's quite a lot of different models, and they all are, in terms of the ranges in the axes, not too different from each other. And what you'll also see is that there's a range of approaches using protein structure, protein language models, MSA, multiple sequence alignment, and that each of these maybe has its own strengths and weaknesses. And one approach would be to try to integrate these different components to produce a better output. One of the methods in the family of methods in protein language models is known as in silico deep mutational scanning. And that means that we replace each amino acid with each other possible amino acid. So you look at our amino acids here, and we just change each one, such as alanine here at start, or we'll change S for N here, or make that replacement. And then with our protein sequence, infer from ESM2 information about this sequence based on its embedding. I move on to the next amino acid and continue this process to estimate how likely it is that this amino acid is viable, and does it increase the fitness of the protein. What can be done then is to revert the change at each site and measure the effect or the site's probability. So just by keep switching the site, we can estimate how viable each site is. So for example, in the SARS-CoV-2 protein, this is from BA.1, and we can change this particular amino acid. So we have an L here after the V for the VLL, and a D here on this side, and we can an F there, whereas the other second site is C here. And we can look at the complex dependencies between sites 2 and 5 here to infer to what extent they're differently dependent. And this is extracted numerically to work out the probabilities of each one. So I might infer that the combination of, for example, F and D and C and L is lower, but maybe the combination of C and D at sites 2 and 5 might be more feasible. We can model newer potential sequences via embeddings and probabilities as well. So for example, what we can do is take existing data from a database, maybe one different from Cascade, extract sequence data from that, apply ESM2 to get the embeddings and scores from that. And then we could do that for an emerging virus as well, to try to get metrics to look at the probability of creating an amino acid change at any particular site, and then plot this as we go along through the sequence. An example of this is openprotein.ai. This is a commercial platform to train custom pre-trained foundation models like AlphaFold2, ASM, and so on. They can do protein function and structure prediction. Um, Another one is Open Protein Python, which is documentation, tutorials, and case studies, um, which also can be applied. Um, One thing to consider is that AlphaFold is not perfect. When it was initially released, the version 1 in 2020, it said solve protein folding, but there's still obviously a lot more to it, and it's another example of commercial companies overselling what they've achieved. Um, so these initial versions used natural language processing and transformers, and version 3 is better because it used diffusion as well. One consideration is that code is not available for it, um, and that's also something that maybe is not doesn't help with transparency of how the predictions are made. Transformer-based models are good at protein structure and long-range interactions with protein inference. And our diffusion model is generative and trying to think about the different options that are present. It is slower than a transformer alone, but it is more accurate, and there's quite a wide variety beyond AlphaFold 3, like RF diffusion, diffusion PLM, and so on. Another example of resources that might be of use is FoldSeq, which looks at tertiary protein structure prediction, showing the nearest of each amino acid to each other. So, for example, we've got queries and targets here. What we can do is calculate split these amino acid sequences into short sequences known as k-mers and compare them to each other and identify ones where there's a match between them and create an alignment between them to create a structural alignment to see their similarity. And then we can um, learn their alphabet by doing atom level modeling of the proximity of each amino acid to its neighbors and then extract information from that. So these are now very, very detailed models. So that concludes this part on protein language models. Please see the demo videos for much more detail on how we can actually apply this using Google Colab in practice.